Hello student, welcome to Raaonline. The chapter is molecular basis of inheritance. This is the part of 12th class NCERT book biology, molecular basis of inheritance. The name of this topic is molecular basis of inheritance. Choose correct option. First one, Friedrich Miescher, discoverer of nucleic acid. Right, he discovered nucleic acid from pus cell nucleus. Mr. Friedrich Miescher discovered nucleic acid from pus cell or destroyed WBC. Pus cell nucleus. He discovered nucleic acid from pus cell nucleus and gave the term nuclein to that one. Gave the term nuclein. Watson and Crick. Yes, they are concerned with the structural demonstration of DNA. They gave a double-stranded double helical model in 1953. They gave double-stranded double helical model regarding the structure of DNA in 1953, and for that they received Nobel Award in 1962. This is the Nobel Award. They received a Nobel Prize in 1962. Wilkins and Rosalind Franklin also helped in the structural demonstration of DNA, where here RNA is written, that is wrong. So third option is wrong, third option is incorrect, because they are also contributor. They also studied DNA by X-ray diffraction method, X-ray diffraction method, and the data of these two fellow, they were also used by Watson and Crick for the structural demonstration of DNA. Hence, third option is wrong. Why? Because RNA is not studied by them, they studied DNA. And similarly, Frederick Griffith, that is DNA is genetic material, he wasn't able to identify. He studied transformation. He studied transformation and wanted to know which is transforming agent, and he was unsuccessful. That rest of the work was done by three fellows. That is, then they are able to describe the DNA genetic material, but not by Friedrich Miescher. So in this question, two options are wrong, 3 and 4, and question says which are the correct options. So 1 and 2 are correct. 1 and 2 are correct, whereas 3 and 4 are incorrect or false. So first option is correct here. Question number 2. Question number 2. Choose the correct with regards to DNA in Escherichia coli. Escherichia coli, bacterial cell. It is bacterial cell. And bacteria are prokaryote. Bacteria are prokaryote. And in prokaryotic cell, true nucleus is not present. True nucleus is not present. True nucleus is not present. Their genetic material is directly present in the cell cytoplasm. This is double-stranded circular DNA molecule. A bacterial cell, double-stranded circular DNA molecule found in cell cytoplasm. DS for double stranded, that is two chains of polynucleotide. Double stranded DNA molecule shape circular. Third, histone proteins are not found. So no histone or without histone protein. This structure acts as nucleoid. Or even we can say genophore. This structure is also called genophore or prochromosome or bacterial chromosome. This is also known as prochromosome or bacterial chromosome. Prochromosome or bacterial chromosome. And in this DNA molecule, number of base pairs, actually double stranded, that's why base pair is correct. First one single stranded, that is wrong. And these are the base pairs of lambda phase. It is a bacteriophase. This is in lambda phase. And the first one single stranded DNA is found in 5 into 174 bacteriophase. 5 into 174 bacteriophase. So the correct answer is fourth. Choose correct about DNA in cell. Choose correct about the DNA in cell. Here the word cell is mentioned, and in cell DNA is always double stranded. DNA is always double stranded. DNA is always double stranded for a cell. If we include viruses, and viruses are non-cellular, in that case single stranded DNA may also possible, but this is the case. DNA is generally always double stranded for cellular structure, and double stranded means DNA is actually polymer of nucleotide. DNA is actually polymer of nucleotide, and here N denotes nucleotide. There is a polymerization of nucleotide. One chain is formed. This is one chain of polynucleotide. If there are two such chain in a molecule of DNA, in a molecule of a DNA, then it is termed as double stranded DNA. Then only it is termed as double stranded, and these two chains are joined together by means of hydrogen bond. So we can say DNA is double stranded now, and these two chains are joined together by means of what? Hydrogen bond is present in between them. So DNA is double stranded, that is two chains of polynucleotide joined together by means of hydrogen bond. Nucleoside. Let's see what is nucleoside. It differs from nucleotide. Actually nucleic acids are polymer of nucleotide. Nucleic acids are polymer of nucleotides. And if we see the nucleotide, then there is a phosphate group, one sugar molecule is there, one molecule of sugar, and one nitrogenous base. All three collectively constitute, this is actually phosphate, P for phosphate, or we can write it PO4, that is, this is phosphate. This is one nucleotide. This is one nucleotide. And the sugar is pentose sugar. Sugar is actually pentose. And this is first carbon of sugar. This is first carbon or alpha carbon is there. Here nitrogen base is present. And this is carbon number 5. This is carbon number 5 where the phosphate group is linked. Phosphate group is present. The sugar plus base plus phosphate collectively known as nucleotide. If we exclude phosphate, in that case only sugar is there. Sugar plus NB stands for nitrogenous base. Then this is termed as nucleoside. This is termed as nucleoside. Refer 11th class biomolecule in which you will get the detail of nucleotides. The sugar plus base portion is nucleoside, and if it includes phosphate then it is called nucleotide. So what is not present? In that case the answer is phosphate group. The answer is phosphate group. And the type of sugar associated with the nucleotide is pentose. Pentose sugar is present. And nitrogenous base either purine or pyrimidine. Either purine or pyrimidine at this position. Purine or pyrimidine is there. Question number 5. The similarity between DNA and RNA. Deoxyribonucleic acid and this is ribonucleic acid. They have similar sugar. Wrong. DNA has ribose sugar. Sugar of DNA is deoxyribose sugar. Sugar of DNA is deoxyribose sugar. That is C5H10O4. Whereas in RNA ribose sugar is present. RNA has ribose sugar. RNA has ribose sugar C5H10O5. That is oxygen deficient ribose is called deoxyribose. Oxygen deficient ribose is deoxyribose. The difference of oxygen only. Then as far as that is DNA is genetic material, both are genetic material, this is wrong. If DNA and RNA are there, in that case DNA is genetic material. This is applicable for the cellular structure. That is, in that case DNA is genetic material but RNA in not that case. Similar pyrimidines, no. DNA has cytosine and thymine, whereas RNA has cytosine and uracil. RNA has cytosine and uracil. In RNA, uracil is present instead of thymine. So how can we say that is they have similar pyrimidine? They have similar pyrimidine is also wrong. So better answer here, that is both are polymer of nucleotide. Both are polymer of nucleotide is the only option. That is, this is the similarity between them. Then read the statement carefully, choose the correct about the double helix of DNA. Double helix, that is there are two chains of nucleotide and they are coiled around imaginary axis. First, it is made up of two polynucleotide chain, right? That's why it is called double. And double stranded because presence of two polynucleotide chain, that is two polynucleotides chains are present joined together by means of hydrogen. And what is double helical? That is both chains are coiled. Both chains are coiled around imaginary axis. Then if we see that is these two nucleotides in a chain, in that case sugar phosphate backbone is present. Sugar phosphate backbone is present, and every nucleotide base is towards inner side. Base is towards inner side. Base is towards inner side. So just a minute. Base is towards inner side. So this is also right. Two chains have parallel. This is wrong. Both are anti-parallel. These two chains are parallel but opposite. So correct is anti-parallel. Correct is anti-parallel. So this statement is wrong. This statement is wrong. Then the the bases in the two strands are joined through phosphodiester bond. This is also wrong. This, just a minute. Bases in two strands are paired with phosphodiester. Paired through phosphodiester, they are paired with the hydrogen bond. They are paired with the hydrogen bond. And the two chains are coiled around in right-handed fashion. Right. That is, helical coiling is from right to left. Right to left. So first is correct. Second is correct. 1, 2 are correct. Then fifth is correct. 5 is correct. And helix pitch, that is 0.34 nm. 0.34 nm. No, it is 3.4 nm. Correct. 3.4 nm is right. So this is also wrong. So final conclusion, that is fourth option is the only correct option with regards to double stranded DNA. With regards to double stranded DNA. Nitrogenous base linked to the pentose sugar. See this one. In a nucleotide, this is pentose sugar. This is pentose sugar. This is carbon number 1, that is alpha carbon. This is carbon number 2, beta carbon. This is third carbon. This is fourth carbon. And this one is fifth carbon. At carbon number 1, nitrogenous base is present, whereas at the carbon number 5, phosphate group is present. The linkage between sugar and base is N-glycosidic bond. This is N-glycosidic bond. N-glycosidic linkage. Whereas the bond between sugar and phosphate is ester linkage. This is ester linkage. This is the correct one. This is first carbon or alpha carbon. This is N-glycosidic bond, and this is the ester bond. This is the ester linkage. This is ester linkage. So seventh question. Seventh question. The correct answer is N-glycosidic bond. N-glycosidic bond. Eighth question. The total amount of DNA present in haploid genome. Yes, it is called C value. Direct question. It is called C value. That is, for example, in human being. Human being are diploid. This is diploid. That is 6.6 into 10 raised to power 9 base pairs are there in 2 sets of chromosome. This is 2 sets of chromosome. Then what about the one set? That is, single set has 3.3 into 10 raised to power 9 base pairs. And in one set we can say this much amount of DNA is present. That is known as C value. The C value for the human DNA, that is, it contains 3.3 into 10 raised to power 9 base pairs. Question number 9. In double-stranded DNA molecule, it has cytosine percentage is 17%. Percentage of cytosine is 70. Here, for double-stranded DNA, Chargaff principle is applicable. Here we can do this question by Chargaff's principle. Chargaff principles is applied. That is, adenine is equal to thymine, that is, they are joined together by means of hydrogen bond. That is, A by T is always 1. And similarly, G pairs with C, G pairs with the C, this is triple hydrogen bond, and G by C, this ratio is also 1. So check it. If C is 17%, in that case automatically percentage of guanine is also 17, because C is equal to G. So CG shares 34%. The remaining is shared by 80. Now ATP 80 base pairs, 100 minus 34, 100% minus 34, that is remaining 66% is shared by 80, and 66% is shared by 80, that is divided by 2, that is A is 33% and T is 33%, and this is also 33%. So the answer of this question is, that is percentage of adenine is 33. Percentage of adenine is 33. ZDNA, unusual form of DNA. ZDNA is unusual form. ZDNA is double-stranded. ZDNA double-stranded. Number 1, it is double-stranded, that is having 2 polynucleotide chain. First, it is double-stranded. Second, these 2 chains are anti-parallel. In this case also chains are anti-parallel. Number 3, double helical. This is also double helical. But helical coiling is different from the BDNA. Helical coiling is left to right. That is left to right. And helix pitch 4.5 nm is right. It is 4.5 nm is right. And number of base pairs per turn for 12, this is also right. In case of BDNA, it is 3.4. It is 3.4, and this is 10 base pairs. So with regards to the ZDNA, all informations are correct. That's why the answer of this question is fourth one. Nucleosome. This is a cell, an eukaryotic cell, and this is nucleus in eukaryotic cell. And within nucleus, chromatin is there. The chromatin is there. And this chromatin is made up of DNA plus RNA, then protein, that is basic protein, histone, and last one is NHCP, non-histone chromosomal protein. The initial packaging of DNA in eukaryotic cell. Initial packaging in eukaryotic cell. This question is from the packaging only. Initial packaging in eukaryotic cell is done by the histone protein. Done by the histone protein. Initial packaging of a DNA. Histone, 8 molecules of histone. These are the 8 molecules of histone. 8 molecules of histone forms core. 8 molecules of histone forms core. 8 molecules of histone, they form core of nucleosome. They form core of nucleosome. And these 8 molecules are H2A, 2 molecules, H2B, 2 molecules, H3, 2 molecules, and H4, 2 molecules. These are the histones and they form core. And this core is wrapped by double-stranded DNA. This core is wrapped by double-stranded DNA molecule. This is dsDNA. Now this structure that a complex is formed, histone plus DNA, that is known as nucleosome. That is known as nucleosome. So histone plus DNA, they collectively constitute what? Nucleosome. And the question is asked only about the core. That is central part, and it is made up of octamer of histones only. And those octamers, that is H2A, 2 molecule, H2B, 2 molecule, H3 and H4, 2 molecules apiece. So this is correct. Second one. If we include DNA, then it will be the nucleosome. But the question is only on core. And the core is made up of histone only. Core of nucleosome is made up of histone only. And number of nucleosome, they are seen in a chromatin just like beads in a string. Number of nucleosome in a chromatin seen like beads in a string. And these nucleosomes, they are joined together by means of linker DNA. That is, in between 2 nucleosomes, what is present? Linker DNA. This is the briefing of nucleosome. And the core of nucleosome is also known as new body. The core of nucleosome is also called new body or V body. Typical nucleosome contains number of bases. See this one. This is nucleosome. It is octamer of histone, H2A, 2 molecule, H2B, 2 molecule, H3, 2 molecule, and H4, 2 molecule. This is the core on which now DNA is wrapped. Take a double-stranded DNA molecule. This is one complete coil. This is second coil. And locked by H1. This is H1. It is not the part of core. This is H1 histone. This is H1. Right? So the question is on this structure. Actually, on nucleosome, double-stranded DNA is wrapped. So How many base pairs are present over this structure? Answer is 200 base pairs, not this one, base pair. 200 base pairs are there. 200 base pairs. See this word pair, that means 400 bases are there, that is 400 bases. See this word pair, here answer is asked in bases only, not pair. So answer is 400 base pair. 13th, choose correct. Histones are organized to form a unit of 4 molecules of histone in a nucleosome. Right. Nucleosome is complex of DNA, RNA and histone protein. This is wrong. Only DNA and histone. Only DNA and histone. Nucleosome is found in prokaryote, eukaryote. This is also wrong. Euchromatin is transcriptionally active chromatin. This is also right. Just a minute. This is also right. Histones organized to form a unit of 4 molecules. Sorry, this is wrong. This is wrong. This is 8 molecule. In first one, 8 molecules. That is H2A 2 molecule, H2B 2 molecule, H2A 2 molecules, H2B 2 molecule, S3 2 molecule and H4 2 molecules. Total number of molecules 8. That's why first one is also incorrect. So this is incorrect, this is incorrect, this is incorrect, and euchromatin is transcriptionally active. Transcriptionally active means that DNA is able to form RNA on its template. For synthesis of RNA on DNA template, synthesis of RNA on DNA template is known as transcription. And in euchromatin, DNA is loose, less condensed. That's why able to transcribe, less condensed. So question number 13, fourth option is right. The opposite chromatin is known as heterochromatin, in which DNA is compactly arranged, condensed is there, and the DNA amount is more in heterochromatin. The amount of DNA is more in heterochromatin. That's why euchromatin is correct, not heterochromatin. In an isolated strain of a DNA, that is double-stranded DNA, this is double-stranded DNA molecule. These are the hydrogen bond. These are the hydrogen bond. And at this temperature, these two chains get separated due to breakage of hydrogen bond. These two chains uncoil and strands separates. This is called denaturation. At high temperature, denaturation occurs. That's why the answer is third. At this temperature, denaturation is seen. Denaturation of DNA. Denaturation. That is, hydrogen bond breaks, two strands separates. Hydrogen bonds breaks. Hydrogen bond breaks. And two strands separates. Two strands separates. On cooling, they reunited. On cooling, they reunited. If temperature is lowered, then again these two chains come close together, establishment of hydrogen bond. The on cooling, these two chains rejoined again. That is known as renaturation or annealing. Renaturation or annealing. And at high temperature, there is a separation of two strands, that is denaturation. That is denaturation. What are the criteria of genetic material? genetic material should be able to replicate. Yes, it's right. Ability to form replica, that is exact copy. That is exact copy. This is the tendency. That is, copy should be exact. Then it should be chemically and structurally stable. This is also right. It should be provide a scope for slow changes or mutation, that is required. This is also right. It should be able to express itself from the Mendelian factor. These are all lines are from the NCERT actually. So that's why answer is 4. That's why answer is 4. That is, all these properties should be there in a genetic material. All properties are, these properties should be fulfilled by the that chemical which is act as genetic material. That is, ability to form replica, that is exact copy. Then it should be structurally stable, chemically should be. Slow changes, yes, they are possible with regards to change in environment. And the last one, that is ability to express itself in terms of Mendelian characters. Refer NCERT genetic in search of genetic material, all are given. 16th question. Which of the following property do not fit for RNA as genetic material? RNA is chemically reactive and structurally unstable. This is also right. Then second OH group present at every nucleotide. Yes, this is also correct. And third, RNA being unstable and mutate very fast, right? If it is single stranded, that's why all these are correct. All these are correct with regards to RNA. It is comparatively less fit for the genetic material, though it is found as genetic material in some viruses. RNA can be seen as genetic material in few viruses, not in all. Few viruses. One of them is COVID-19 virus. It has also RNA as single stranded, RNA as genetic material. And if RNA is single stranded, it has the tendency to mutate fast. That's why still the medicine or vaccine of COVID-19 is not obtained or not discovered, because the changing behavior of its nucleic acid, that is RNA. 17th question. Some viruses do not have DNA. I have already mentioned that in case of viruses, see this one, viruses made up of nucleic acid plus protein, this is common structure, and envelope in them optional. Envelope in them is optional. The nucleic acid of virus, either DNA or RNA, either DNA or RNA. I mean to say, if DNA is there in a virus, then RNA should not be there. If RNA is there in a virus, then DNA should not be there. That is, both DNA or RNA and RNA can't be seen in one virus. So viruses are of 4 categories with regards to DNA, genetic material. That is RY1 containing, that is single strand RNA containing, R by 2, that is double stranded RNA containing, D by 1, that is single stranded DNA containing, and D by 2, that is double stranded RNA. There are 4 options are there. So let's see what is suggested if they don't have DNA in that case. They are non-infective. This statement is wrong. RNA as the genetic material of these viruses, this is the absolute probability. Yes, I have already told only in viruses either DNA or RNA is present as genetic material. That's why second option is correct in them, and that is either are single stranded or double stranded. This is the central dogma of molecular biology. This central dogma was given by FSC Crick. Who gave this central dogma? FHC Crick gave this central dogma. And see this one, that is DNA. This arrow says that DNA is able to synthesize DNA itself. That is, first A is replication. This is replication. Ability of a DNA is to form its own exact copy is replication. This is the unique property of a DNA, that is to synthesize itself exactly. Then DNA to RNA. Here the option is mentioned D. See close look here. This is known as transcription. This is known as transcription. Synthesis of RNA on DNA template. Synthesis of RNA on DNA template is transcription. Then what is about this one? RNA if synthesized DNA on its template, that is reverse process of the previous one. This is reverse transcription. This is reverse transcription. It is seen in some viruses only. This is reverse transcription and is seen in some viruses only, not in all. Those viruses are called retroviruses. Those are called retrovirus group. Those are called retrovirus group. And the B part, that is RNA to protein, that is information contained in RNA, is translated in the form of protein over ribosome. So the last part is translation. The last part is translation. So let's see which one is correct. A replication in first and in third and in fourth. So second option is already rejected because of A is a replication. Now come to the B. B says, B is actually mentioned here for the confusion. So B is a translation. B is a translation. So now in this case this is rejected, that is question 1 rejected, option 2 is also rejected. Now in 3 and 4 B is mentioned as translation. So we have to move again, then come to the C. C is reverse transcription. C is reverse transcription. Now this matches. This option matches. So final answer, that is C, not D. That is C, not D. D is a transcription process. So this question, that is third option, is absolutely correct. What is the replication? Synthesis of DNA on DNA template. That is ability to form DNA from DNA. This is called replication, and it forms its own exact copy. And synthesis of RNA from DNA, that is synthesis of RNA on DNA template, is transcription. And synthesis of protein from information contained in RNA, this activity takes place over ribosome, that is translation. And if RNA is able to synthesize DNA on its template, this is reverse transcription. This is reverse transcription. So question number 18, answer 3. Question number 19, reverse central dogma Ross sarcoma virus, right. Who studied Taman, Baltimore, Delbecco? Answer right. RNA dependent DNA polymerase is called needed for the reverse transcription. This is also right. Then reverse transcription is useful for the formation of cDNA, that is complementary DNA. This is also right. So my answer in that case is 1, 2, 3, 4 all are right. Synthesis of DNA on RNA template. DNA on RNA template, that is reverse transcription, right? Then enzyme needed, that is reverse transcriptase, which is also called, already mentioned, reverse transcriptase, reverse transcriptase, reverse transcriptase, that is RNA dependent DNA polymerase. And what is cDNA? That is DNA from RNA and RNA from DNA. If RNA synthesizes DNA, then definitely it is a copy of a DNA. This is a copy of a DNA, that is they have same informations of protein in them. So that is cDNA or complementary DNA. It's a comparatively longer process to explain. Better it will be dealt in what portion, that is during transcription. We will study in detail. Element used to label the bacteriophage and varying binders experiment, that is Hershey and Chase. Hershey and Chase proved that DNA is genetic material. DNA is genetic material. This was proved by Hershey and Chase. They experimented over T2 phase, bacteriophage. T2 phase, that is bacteriophage. In bacteriophage, this is proteinous covering on head and this is tail, and in tail 6 tail fibers are there. 6 tail fibers are there with 6 spikes. And all these structures are protein. All these structures are of protein. And DNA is found only in head region. DNA is found in head region. Protein is made up of amino acid. Protein is made up of amino acids, and some amino acids are sulfur bearing. Some amino acids are sulfur bearing. So protein can be, that is this proteinous code can be labeled with sulfur 35, that is for the protein. This is for the protein labeling. Sulfur 35 is used for the protein labeling. And DNA has phosphate in their nucleotide, phosphate, sugar and nitrogenous base. So phosphate portion is found in DNA. So for the labeling of DNA, P32, that is 32. For the labeling of DNA, okay, DNA labeling. What is used? P32. And question, what is the question? That is for the labeling of DNA. For the labeling of DNA. For the labeling of DNA, this is the only question. So in that case, answer is P32. That is P32 form of phosphorus used by Hershey and Chase in their experiment. Michelson and Stahl's experiment. Michelson and Stahl they proved that method of replication of DNA. Method of replication of DNA is semi-conservative. Semi-conservative. They experimented over Escherichia coli bacterium and they used 15N. that isotope of nitrogen, but it is not radioactive. It is not radioactive. N15 is not radioactive. This is correct. It is not radioactive. So here this N15 is supplied with NH4Cl in culture medium. If it is added in the culture medium, then Escherichia coli is added in the culture medium. E. coli multiplies, that is, it undergoes division. During the division, DNA replicates, and the further replication of DNA, new nucleotides are needed. These new nucleotides are incorporated with the 15N instead of 14NM. That is, E. coli received 15N type of nucleotide from the culture medium. Then only two different types of nucleotides can be identified. The older one of N14 type and the newer one is of 15N type. 15N type. So it is supplied with what? That is NH4Cl, and in this case N is of 15N type. Assuming 50 heavy DNA. 50 heavy DNA, that means both strands having 15N type. 15N, both strands are 15N type. And the replication is done twice in a medium containing N14. So let's check one DNA molecule only. This one DNA molecule, it has 15 and 15 nitrogen on both the strands. The medium is now N14. Medium is N14. In division number 1, two DNA molecules are produced. And see them. The blue one indicates 14, the red one indicates 15. Blue one indicates 14 and red one indicates 15. This is generation number 1. Come to the second generation. In second generation, this is the separated strand of this one, and this is the separated strand of this one, and the blue one, this is also a separated strand, this is separated strand, and the medium contains N14. So this strand forms N14 type, this strand is 14 type, this strand is 14 type, and this strand is 15 type. Sorry, 15-14. So out of 4, 2 with semi or half heavy DNA, semi or we can say half heavy DNA, whereas 2 DNA light DNA. This is out of 1. And here 50 DNA molecules are there. So first of all, 50 DNA becomes 100 in second generation and 200 in third generation. And apply this principle, that is 50% DNA are light, that is 100 DNA are light, 100 DNA molecules are light, and remaining other 100, they are half heavy. So check the answer now. 100 half and 150 not possible. Here the number is different. 100 half heavy and 100 half light. No half light DNA is present. 50 heavy, 150 no. 50, 100 no. That is second one is closer. That is, we can say 100 half heavy or half light, and remaining 100 light DNA. That is, both the strands are labeled with N14 in this case, N14 in both the strand in light DNA case, whereas in half heavy DNA, one strand is of N15 and one strand is of N14, and this can be done only in this way, that is, take one DNA molecule and first of all get converted into 4 DNA molecule after 2 generations, and then apply this particular concept on the 200 DNA molecule. So answer is 2. Answer is 2. Thank you.